Okay, hello to Propellicure's video series where we highlight innovative research into the causes of Crohn's disease and potential treatments. Today, we are joined by two researchers from Australia, Dr. Samuel Forster and Dr. Edward Giles. Dr. Forster is a CSL Centenary Fellow and Associate Professor at the Hudson Institute of Medical Research. He received this highly prestigious fellowship to advance his work using the bacteria of the human microbiome to treat inflammatory bowel disease. He is also the Chief Scientific Officer at Biobank, a clinical stage biotechnology company with a mission to treat and prevent disease by restoring the gut microbial ecology. Dr. Forster is a world-leading microbiome scientist who has pioneered key research in metagenomics and anaerobic culturing of bacteria. He's published papers in Nature and Nature Biotechnology detailing culturing of unculturable organisms. Dr. Forster's research focuses on gaining an understanding of the microbial communities that live in our bodies, bringing together experts in microbiology, immunology, and computational biology. His team has developed a new way of taking gut samples and looking at the bacteria within normal and damaged gut tissue. Importantly, they have discovered and are testing promising bacteria that could be used to treat and/or even cure IBD, which is music to our ears. Additionally, Dr. Forster aims to identify when bacteria represent a risk and should be removed, and when they could be beneficial and should be added to induce a positive immune response. Dr. Giles is a clinician scientist and senior research scientist in the regulation of interferon and innate signaling research group at the Center of Innate Immunity and Infectious Disease. He is a fellow of the Royal Australasian College of Physicians and was awarded his PhD in type 1 interferon signaling in human intestinal T cells from the University of London in 2015. He researches the mucosal immunology of inflammatory bowel disease, particularly the role of type 1 interferon and their influence on the adaptive immune system and microbiome in the intestine. He is also a consultant pediatric gastroenterologist at the Monash Children's Hospital, where he is lead for pediatric IBD. So thank you both for accepting to discuss your research with us. And now I'll hand you over to Kathleen Pickney and Lisa Kelly. Kathleen is a patient and has been struggling with Crohn's for a very long time. And Lisa has a son who has also been struggling with Crohn's for a very long time. And my son's been also struggling with Crohn's for a very long time. So there you go.
Thanks, Annabelle, and thank you, Dr. Giles and Forster, for meeting with us. We're really excited to dive into your research on the microbiome. There's so much out there on the microbiome right now and so much to learn. We were really excited to learn about the new findings you guys are discovering when it comes to the relationships between the bacteria. But can you first dive into the new technique you're using to study the gut microbiome, where you're taking the samples and using the gut on a dish?
Yeah, I can perhaps start on that. So fundamentally, what we've been focused on is a couple of ideas. So first of all, a lot of the microbiome research that we're doing has been focused on fecal samples. And that's a cross-section of everything in the gut, right? So we know Crohn's disease doesn't affect just, the whole gut inflamed the whole way through. We know that there's patches where you get inflammation. And so we focused on being able to understand the precise microbial community, so the bacteria and the cells, your own cells, and how they interact at particular sites. And there's multiple ways we do this. So first of all, we have to collect the samples. So we take biopsy samples and measure what's actually existing in the time when the disease is at its worst, but also at the times when the disease isn't actually as bad. And then what we can do is we can grow these individual bacteria, and we can also grow the cells from the patient, and we can see how they're interacting. So we can understand what inflammatory pathways are being activated, how the different bacteria are interacting with each other. And to do this, we need a number of techniques. So we need to be able to computationally, we sequence the genome. You might have heard of the Human Genome Project, but each of these bacteria have their own genomes as well. So we can measure those genomes. We can understand what they might do, how they interact. They all live as a community, just like we live as a community, and some people get along, some people don't. The bacteria live as a community, and some of them get along and some of them don't. And depending on these interactions, they can produce certain things that can then affect our own guts and cause the inflammatory state that can be detrimental. So if we understand how they work together and how we can potentially change them so they work together in a better way, that gives us an opportunity. And as you mentioned, the gut on a chip technology allows us to replicate some of these interactions in an experimental setting. So we can say, well, what would happen if we changed the bacterial composition? What would happen if we put a different bacteria in there, or if we removed a particular bacteria? So that's the opportunity, and that's really the area that we're pushing into and trying to understand. The other thing we have to remember is that all of the bacteria that we're carrying it isn't a static thing like a genome. Every day we're exposed to these bacteria. We know, I mean, we have these times where we'll eat some food and we'll get a detrimental effect. I mean, everybody can think of a time where they've had some kind of food poisoning or other response to eating a bad meal. But how many people think about, I've had a good meal today, I've got good bacteria and it's actually beneficial? The reality is we're seeing these bacteria, these beneficial bacteria every day. Perhaps some of our changes in lifestyle are affecting which ones we pick up and which ones we don't. But fundamentally, it's a dynamic community that has the opportunity to keep changing. And some of those states are beneficial and some of them are detrimental. And what we're trying to do is tease that apart, understand, particularly at the times in Crohn's disease where the disease can be the worst, understand why the bacterial community is existing as it is, and is there an opportunity to change that community such that we can reduce or completely remove the effect.
That's very exciting to hear, specifically that you're able to test the bacteria and see maybe what's working with it and what's not. Do you suspect that the same bacteria are at play across all Crohn's patients, or is the microbiome heterogeneous and therefore will need to be individualized per patient?
Ed, do you want to answer that one?
Yeah, no, you go first, Sam. But I know what you're going to say. So, but I'll let you take it.
Yeah, I mean, I think the reality is there's a few factors in the microbiome research space that are kind of interesting. So traditionally, people have said there's quite a stable microbiome community, but that's measured with fecal samples. So we know that there are changes associated with you have a meal, there's more nutrients there, certain bacteria like that. So we have this trade-off between stability and then everybody being different, which is the other element that people talk about. But there's still shared functions and there's still subgroups of people where they have similar effects. So I don't think it's going to be a case where we have this one probiotic or one live biotherapeutic that you put in and everybody's suddenly cured. But similarly, I don't think it's going to be a case where for every individual you have to give a perfectly designed combination. We know, for example, that with fecal transplant in IBD, generally the response rates are about 30 or 40%. There's no targeting there in terms of the composition you're putting in. So that suggests it's not everyone, but there's a proportion where there's a potential intervention. And if you had a targeted intervention, perhaps you could then divide that further and say, okay, we for a particular patient, we can measure their microbiome, we can understand how that community might be interacting, and then we can say this is a combination that's likely to restore the healthy state, or at least reduce the extent of the symptoms that the patient's experiencing.
You mentioned FMT or fecal transplants. Can you talk about, first, before we dive into that, biotherapeutics as a whole? What are they and what place do you see them taking place for IBD in the future?
Yeah, I mean, I think it's a very broad area. I mean, Ed's probably better placed to comment on the precise details than I am, but fundamentally, it's that any targeted intervention where you're trying to change the microbiome could have a therapeutic benefit. So that you could think of that in terms of prebiotics, probiotics, light biotherapeutics, FMT. So whether it's you put in a compound that's going to change the growth of the microbiome, so encourage certain species to grow or perhaps remove certain species, whether you put in a live bacteria or other microbe that's going to change the community because it's going to be there and be a part of that community. and that would include fecal transplant, even though it's not done in a targeted way, or whether you say actually there's a particular component of the bacteria, so maybe part of its cellular surface or something like that, that induces or changes the immune response, and you don't need the bacteria at all. You just purify that component and put it in, and that gives you the same overall effect. And it's possible that any combination of those could be beneficial. At this point, it's really about getting a better understanding of the precise microbial composition. So if we can get that understanding of, for this subgroup of patients, as you described before, if we can say this is the composition of the microbes and this is why it's detrimental or this is why it's beneficial, then we can start to say, well, how are they interacting together and how can we then change that community? And that's really what we're focused on. How do you change the community in a targeted way rather than, as you said before, with the fecal transplant, where you're just putting in a mix and hoping that that composition will change something in a beneficial way?
I have a question. So, Crohn's, of course, there are all sorts of different symptoms. And some people have just Crohn's and ileocrohn's, where the other people have colonic Crohn's. So does this, what you're talking about, is it specific to one type of Crohn's, or could we use this broadly?
Perhaps I should come in to give Sam a bit of a break, Sam. So I think that's a really key question, Annabelle. And I think that, one of the things that as a clinician that I've come to understand about Crohn's disease over a number of years is that, any conditions that's still named after an old white guy a long time ago probably means we don't understand that disease at all, and that it's probably a number of different diseases. And we classify that as clinicians, and you might have heard of these classifications types, but they're not particularly helpful in terms of guiding treatment or even prognosis. We're just not very good at that. Mm-hmm. And I think that demonstrates a fundamental lack of understanding, really, that there's a gap in our knowledge here. And it's not certain, but we would hope that by having a more site-specific approach to understanding the disease, and with Sam, particularly from a microbial and a sort of host bacteria interaction perspective, understanding that in that particular way, that we can have a greater understanding of why there are these differences. And then that can then move to, more predictive models and interventions, really, because of that. So I think that there's a sort of, and I think this has really been a barrier for Crohn's research, possibly until quite recently, that, you might think that you can get 100 people with Crohn's disease and study them and try and find some relationships. But if you've got 50 different diseases in there, or maybe not quite that many, the numbers of people that you're studying in those groups are really too small, and you're probably making false associations. Whereas what we can do, or Sam's technique can do, is we can sort of take away all that and really just do a computational analysis of the relationship between the inflammation and the damage of the bowel and the associated bacteria, and see what those relationships are, and not include whether someone has Crohn's disease or otherwise in that analysis. So I would see those diseases, while there's probably some degree of spectrum in there, they need to be thought of separately.
Okay, makes sense.
And as you specify FMT more and look at these subtypes, if microbiome therapy like FMT becomes effective, do you see this as a therapy that needs to be taken or done for life, or could it potentially repair the microbiome initially so that treatment can eventually be discontinued?
Sam, you take that one.
I mean, I think it's an unknown, obviously. The hope is that we can create a situation where the microbiome is so stable that it won't change back to the detrimental state. The likelihood is most people won't want to adhere to a very rigorous diet and all the controls on their life in terms of maintaining that microbiome. They'll be exposed to other microbes. So it probably will require some degree of introduction of further bacteria, but ultimately you would hope that that would be done with a tablet every day or something like that, in a simple way that really doesn't have any major impact on the day-to-day life. Obviously, though, it's still early days from that perspective.
I'll come in on that. I think another thing that Sam's group and we look at, I'm a pediatric IBD specialist, and I've trained in Australia and then spent a long time in the UK as well. And we use a lot of exclusive enteral nutrition as our first line of therapy for pediatric Crohn's disease, and that's being used more in adults as well. And I think one of the things that we think about is this so-called paradox of, we know that at least in the stool and the fecal samples, that the diversity of the microbiome is reduced when you have Crohn's disease, but we also know that if you take enteral nutrition, you really reduce the diversity, so you have much fewer bacteria in lots of different measures when you're on that diet, and yet we know it heals the bowel. And so we're sort of trying to understand that slight paradox of those things, and understanding as well that at least with ulcerative colitis, this idea of fecal transplant and replacing the microbiome can be helpful, and understanding that the link between the diet and the microbiome and that inflammation is really important. And at the moment, the only diets that seem to be effective in Crohn's disease really are so restrictive that they're unfeasible in anything other than the very short term. But perhaps if we can understand that relationship as well, and the nutrients that supply in the different groups of patients, we can do some combination of replenishment and a less extreme dietary intervention that might be feasible, particularly if you can cohort that with a particular type of patient and their microbiome ecology, for want of better words.
Mhm.
And Dr. Giles, that brings me into your research on hydrogen sulfide. It was kind of talked up until now that maybe Crohn's patients have an excess amount of bacteria, but you found something different. Can you explain that?
Yeah, I mean, I think that this is really about the paper that Sam led and I was involved in, Vanessa Marcellino's paper looking at metabolic exchange, so understanding really about the communities of bacteria and how they work together. And I think that once you start to look at things in a community structure, you often find quite different results than that more isolated individual approach. And I guess what one of the key concepts from that work is this idea of producers of metabolites. And when it came to the Crohn's part of that paper, which was a major feature, really the strongest finding was in the hydrogen sulfide, and then consumers of that metabolite, and seeing a disjunction in certain cases, and in that case, Crohn's disease. So really it's about trying to understand the metabolic balance and how certain metabolites, so hydrogen sulfide being something that can be both produced and consumed within the bacteria, and how that difference can then be part of the problem that might then contribute to that inflammation. But again, a lot of that data is really from fecal samples, and we need to understand at a more site-specific level whether that's the case, and using some of the experimental models that Sam's looked at, trying to understand that further. Sam, do you want to comment a bit more on Vanessa's paper?
I suppose the key element there, so the sort of historically we've known that excess hydrogen sulfide can be a problem for Crohn's disease patients. The whole study highlights precisely the concepts we've been talking about, in the sense of what we've been able to do is understand the precise interactions, as Ed was saying. So understand that these bacteria are sharing nutrients, sharing metabolites. And one of the effects that we've seen in these Crohn's disease patient samples is actually that it's not that there's too many species producing hydrogen sulfide and they're going up, and there's more of them, so there's more hydrogen sulfide. What it seems to be is it's actually a loss of the species that would normally be consuming that hydrogen sulfide and maintaining the balance. So it's sort of counterintuitive in a way. You think more hydrogen sulfide, there must be more species producing that hydrogen sulfide. But actually what the data suggests is that the production side of that equation doesn't change at all. It's the species that would usually be using it. They're not using it in the same way, or they're not there anymore to use it. And therefore you get an excess amount. And then that excess amount is detrimental because of the effect it has on our own cells. So it's this you don't necessarily have to have a preconceived notion of what's going on. You you can focus the analysis on a part that's relevant and say, well, how does the composition explain that interaction? And that's exactly what we've done here. Obviously, there's lots of metabolites in these systems. There's lots of interactions. But part of the framework, and this is more broadly than Crohn's disease, but part of the framework that we've developed starts to allow us to focus on, well, what are the key metabolites in a particular state? And that could be, if you imagine there's one metabolite that all the species in the community need to exist, and there's only one species producing it. Protecting that one species that's producing it is really important. Otherwise, all the other species suffer from not having it. Similarly, it might be a case where we have this situation where there's lots of species producing it and lots of species using it. And then it's a metabolite we don't need to worry about as much, because even if one of those species that is producing it is lost, there's others that are likely to still be fulfilling that role. And you can obviously imagine that in reverse in terms of the consumption, which is the hydrogen sulfide side, where if you have lots of species consuming it, then if you lose one of those species, it's less of an issue because the others are just happy there's more of it for them to use. If there's one species using it and you lose it, then suddenly you get a buildup of that, and that might be detrimental. And so that's the concept and why we're taking this ecological approach to understanding the communities. But it also allows us to take an ecological approach to how we then change the communities. And that's obviously key from a therapeutic intervention perspective. If you can imagine, coming back to what you were saying about subgroups of patients where they have similar disease, if they also have similar functional interactions like this, we can understand those interactions, and then we can go back and say, well, this is the group of species or this is the function we need to restore that would be beneficial for those patients.
Do you also believe that we should be taking into consideration the fungi that are within our bodies and not necessarily just the bacteria? And can you give an overview of how they play into each other?
Yeah, so I think we have to remember it's very easy to talk about the bacteria. That's certainly where the majority, though not all the research focus has been. And that's largely for technological reasons. The measurement and the detection growth of these species is easier. There are other components. So there's the fungi, as you mentioned. There's in some individuals, though more rarely, there's other parasite type organisms. There's also viruses, both viruses that affect us, but also viruses that affect the bacteria. So there's all these interactions, and we have to take these these full community dynamics into what's going on, because it's not like the bacteria and the fungi are like, well, we're different, we don't talk to each other. There's all these metabolic interactions are just as likely to be occurring between each of these components, and that's really where the focus and the understanding needs to be. If we've gone from if we think about what's been achieved over the last decade or so, it's amazing. We've gone from a vague understanding of the community structures to being able to having these huge cohorts of patients with Crohn's disease where we've got sequencing data, so we've got an idea of what composition's there. But we've now gone to, well, we can grow these things, we can start to functionally understand these things. And the next stage is to say, well, let's go beyond bacteria. Let's bring the full community state in, but also consider the patient that's sitting around this community in terms of how the immune system is interacting with these microbes, but also how the behavior of a patient, whether that be diet or lifestyle, etc., can add to or remove the ability of the new bacteria or new microbes to colonize. So it's bringing the next layer of this research, and the really exciting space in this research is bringing all these elements together such that we can get these functional understandings that we're talking about today, where it's not just a case of, well, here's a cohort, if you're part of this cohort, you're more likely to carry this particular group of bacteria. It's functionally, we understand how these communities interact, how they work together, how that has a beneficial or detrimental effect to us. And with that understanding, we can then start to go in and say, well, this is the best way to change it, or this is a way we might be able to have a beneficial effect.
And Dr. Giles, could you talk a little bit about this protein? Forgive me for butchering the name, but interferon epsilon. What role does that have in the microbiome?
Sure. So this is another sort of separate but sort of
Okay.
exciting project that I've been leading in the lab alongside the sort of mentor of both Sam and I, called Paul Herzog, who actually discovered this protein. So this is part of a family of proteins called type 1 interferons, which are really important in protecting the body against viral infection, discovered in the 1950s. But there's lots of the different members of this family, but epsilon was only discovered at the Hudson Institute sort of now perhaps a little bit over 15 years ago, and was initially found to have an important role in the female reproductive tract. And I've been leading work on it in the gut, because it's what's unusual about this protein is only found in the sort of lining cell layer. So this is called the epithelium, which is just a single layer of cells that lines parts of our body. And obviously what I'm interested in is that lining of cell in the gut. And so this has been shown in some animal models at this stage being critically important for maintaining the integrity of that barrier. And we often talk about a leaky gut in inflammatory bowel disease and other conditions as well. And we can show that without this protein there, you get this leakiness, and this really affects vulnerability to damage. And we've also been able to show through our human work that it's present in children and adults with in the intestine. And we're sort of working to look at understanding how the microbiome interacts with this, because clearly we need to look at both sides of this coin. And I think that's the sort of overriding story of the work that Sam and I try and do together, is really understanding that interaction and what holds things together. Because there's a lot of work's been done on the genetics of inflammatory bowel disease, and while we're understanding more and more with children that there can be a very small proportion of children with genetic disorders that sort of mimic Crohn's disease, the majority of cases that's not really the case, and that the power of the genetic influence is generally quite small. But there is some setup, and there's often a setup early in life. We know that there are events in your first year of life, and as a pediatrician, I'm really interested in this, that sort of predispose to some trigger, but that trigger often doesn't come until the 20s for patients with Crohn's disease. Now, I look after patients where that happens earlier, and it must be perhaps, or it's logical to think there's a stronger environmental effect. But what is it going on at that barrier level that's breaking that down, that we go from coping with the billions and trillions of organisms of all sorts, bacteria and otherwise, and maintaining that balance at that one cell level, and then that being disrupted. And so I see perhaps a bit like some of our findings that we've published in our work of the microbiome, looking at one protein is hopefully an attractive target for therapy, and this is true because it's just in that lining, that it might be a safe treatment, because it doesn't necessarily have the effects through the whole immune system like other treatments do. But it's also about really understanding that interaction with the microbiome and trying to really understand it at that site level, what's actually going on that helps us maintain balance away from disease.
And just putting all the dots together, what can you lay out the exact, or what you see future opportunities are to translate these findings into therapies? We know about the potential of FMT, but are there other similar therapies that you see?
Absolutely, and that's ultimately the goal, really. I mean, I think we work very hard to have a better fundamental understanding of particularly the bacteria and how that relates to the immune system. And that's because we're trying to get away from being too colored by past thoughts about labels of Crohn's disease, to really understand that fundamentally. But the goal of that understanding is to move forward to translate that. And what's appealing about this line of discovery type science is that the path to translation, we think, is relatively clear, because we're looking at products, whether that's live bacteria or their metabolites, or even pathways, as Sam alluded to earlier, such as epsilon that I'm looking at, that interact with those things, that can be targeted, and that treatments already exist. Or if they don't exist, we know that these bacteria exist in a healthy state. So safety in terms of designing and then beginning sort of what we'd call a phase 1 early trials should be relatively straightforward. I say optimistically, but in the sense that if we know that these are already safe in healthy people, you're not giving something that's foreign. You're actually giving something that's known to be tolerated. And so really what we're looking at is trying to understand that so we can move quickly. And again, as Sam says, we're hopeful that this isn't too individual, and our data really, at least initially, supports that it's more about understanding groups of patients in a different way that's defined by both their microbiome, but also the way their microbiome interacts with the specific site of their bowel, and then being able to target that initially in a way that interferes with that host microbe interaction in a way. So I think it is a really hopeful time, because I think that once we can get through this period, and the reality is that the data is coming really quickly from many groups around, and I think there are other groups doing this. And so I am relatively optimistic that we can then move to looking at treatments in a relatively short time frame, within numbers of years, not decades.
Ooh, that's good to know. That's very helpful. Because I've sort of, I don't know, I've gotten to the point, it just seems everything is so slow. And I think I talk for a lot of people. It just, we feel like, and many times that the urgency isn't, we're feeling the urgency, but, and maybe some of the researchers are, but it just seems like the powers that be, it's just so slow in publishing.
Well, I think, speaking more as a clinician than the scientist, there have been new therapies that have come online for patients with Crohn's disease. In the early part of my career, and I'm around sort of peri-biologic, if you know what I mean. Infliximab was around for Crohn's disease when I really started working in this space, but not really for ulcerative colitis. And so we can see that the numbers of treatments have really increased. But I guess what we haven't seen is really a change in approach, particularly from the pharmaceutical companies yet, although I think that is also changing as they're understanding that we do need to be a bit more targeted in our patient groups, and that there is a pivot in the commercial space to understanding the microbiome and that interaction as well. And I think that what's allowing this optimism on my part, I think, is probably the technological advances that Sam and others in his field are doing, not just in our sampling, which is really important for the work we do, but computationally being able to handle these large data sets in a way that just wasn't feasible a decade ago, because the complexity is undoubtedly enormous, and that is limiting. Even now, it's limiting. Some of the analyses that we do still take days running through very high-powered computers, but that sort of technology is improving all the time, and it does, I think, give some hope to at least an acceleration in the speed of our understanding that then allows the movement into something that's more practical use to patients.
Yeah, I think that was something that really attracted me to your work is Dr. Forster, is the fact that you bring in all of these different aspects of it. You have people who are immunologists and microbiologists, but also computational parts in it, because it is. From the little that I know of it, as I'm not, obviously not a scientist, it just is very complicated, and all of the different articles that come out, it sort of seems like it's one is there, and then one is over there, and then one is over there, and nothing ever seems to be brought together in a full comprehension of what's going on. And I think that's been probably a hindrance in advancing, because it has not been enough collaboration, I think, to also to make this all one piece. So, yeah.
Yeah, I mean, I think we have to, I do agree with what you're saying. I think we have to remember, though, all of these fields are advancing so quickly that it's a case of keeping the best technology from all of the fields, talking to each other and bringing it together. And as you say, that's obviously what we're trying to do is have, you want someone who's an expert at modeling metabolic communities sitting next to someone who's an expert at the detail of growing the bacteria, sitting next to someone who's an expert who's at understanding the host response and the clinical side. And if those people are talking to each other and focused on how do we understand what's going on and how do we intervene to change it for a benefit, that's where we can really make the progress. And that's really where we see the exciting opportunities.
Yeah, and I'll just add on to that, Annabelle, just to, I think coming back to Sam's own work and his role in this, when we think about the microbiome, and there's been a lot of research in that space over the last 20 years, say, maybe slightly less, but really this understanding of the anaerobic bacteria, so the fact that our gut is this space that cannot tolerate oxygen for many of the species in there has meant that there's been a great limitation on that until really the Nature paper of Sam's from 2016, and then that was really on a very small number of healthy individuals. And this has been rapid expansion. And even in our own work, we're discovering new species and strains at quite I would say almost an alarming rate. But the technology is there to do that. But I feel like, this was an area that we just couldn't understand for technological reasons until relatively recently.
Right.
And so I think that I often describe it's like a room in the house that you didn't even know was there. And you're suddenly like you've opened it up and it's, there's a lot to go through. And lots of groups are doing that now. But getting them all together when it's really this new space is a challenge, and the system isn't always set up to support that perfectly, but it is to a degree. And that will come. And I think that, I think it's sometimes a newer space than people realize because people talk about it so much.
Right. Do you guys have any, just sort of inclination in a way about, because I mean, the cases of Crohn's disease, of course, as we all know, is going up all over the world, but particularly in right now in developing countries. And it's sort of plateaued a little bit, if we can call it plateau, in countries like, the United States and Europe and Australia. But now Asia's and then South America, we've seen more incidences of it. Do you have any kind of even if it's not scientific, any idea of your own of why you think this is happening?
Sam, do you want to go first, or what do you want me?
You go first.
I mean, I'm sort of really interested in this sort of early life events, and I think that there's been a lot of talk about diet, and the sort of Western diet. There's certainly something about the Western lifestyle that I think is clearly important, but I'm not 100% sure that's necessarily a dietary intervention, or at least not in the sense that we've seen, because I think even in those large studies from the US nurses studies, etc., it's very easy for diet to be confounded with other elements of lifestyle, because people who lead healthy diets tend to do other things as well. And so, but I am persuaded by things like the antibiotics in the first year of life and what that does. But I wonder whether it's also about exposure to particular viruses and bacteria in that early part of your life. So in Western cultures, we might send our children to childcare, for example, and be exposed to more viruses. In other conditions, maybe there's exposure to other microbes earlier in life that sort of sets somehow your tolerance for later insults. But there's got to be something that sets your trigger point, and then there's also got to be a trigger, because we don't spend a whole life with inflammatory bowel disease, but there's clearly something in the genetics. And while that hasn't been helpful from a treatment perspective, that has helped us understand some of the immune pathways from the genetics. and then something in the early life, and then a sort of later trigger. And I think that, but I think this is a very difficult thing to completely understand, because for the majority of our patients, there's a 20-year time lag to kind of understand that. But that would be my theory, something about exposure to microbes and different sorts of microbes early in life, changing your immune set point, if that's a term that could be used. Sam, you got anything to?
Yeah, I would agree with
down?
No, I would agree with what you've said there. I would make one other point that I think we forget a lot. And, obviously, I from a microbiology background, the majority of the microbes out there are actually not things trying to kill us. Yet most of the Western activities are about killing microbes. So, we want to, rightly so, we want to stop infections, we want to kill pathogens. But actually, everything we do is about cleaning things, stopping the spread of microbes, stopping the movement of things. And actually, if we say, well, for our gut, for example, there's hundreds, if not thousands, of species. A handful of them can be detrimental, can be disease-causing. Yet we're putting in place these things that stop all of them spreading between individuals, stop the majority of them spreading between individuals, prevent that recolonization. We know a healthy ecosystem is about the diversity, is about movement of these microbes. So perhaps it's part of coming back to what Ed was saying, part of that reason we're getting these different exposures at different times of life is about single-minded focus on killing every microbe that we come across. And it's a really difficult balance. It's not like we can suddenly say, well, forget about antibiotics, forget about treating pathogens. But we've got to somehow think about, well, how do we keep the ones or help the ones that are benefiting us, such that we can improve our exposure overall. And it's certainly not something that there's a huge amount of evidence to support, but just on first principles, you can see how these interactions could be associating. And it's something that's very difficult. The space of how do microbes move through the environment, the health-associated ones, is an area of active research that's as new as all of these other areas of research that we're talking about. And if you think about it, if you're tracking pathogens, if you think about how COVID transfer worked, it was very easy to identify the people with COVID because they had symptoms, and you could then test them and say, yes, well, that's COVID, and so forth. If you're saying, well, you get exposed to a microbe today that's going to be beneficial for you in 20 years' time, how do you, other than testing everybody who could have come in contact, how do you test that? And then multiply that, rather than just one pathogen, by thousands of bacterial species. You can see how that becomes a very difficult game to understand on a large scale. But if we can understand some of these fundamental principles, and we've got decades, if not hundreds of years of understanding how to measure pathogens moving through the environment, how to measure how these pathogens affect a particular individual when they get infected. If we can apply all of that knowledge of how to understand those things back to the commensal microbes, that whether they're beneficial or detrimental in a particular state, that suddenly unlocks a huge amount of opportunity. And obviously, because as we've kept coming back to, because these things are naturally occurring in people, as a therapeutic intervention, they're actually quite a promising opportunity. We have to remember, each one of us in our guts has 2 or 3 square meters of space where there's supposed to be a large number of bacteria interacting with our immune system to modify the way that we're experiencing the outside environment. And at the moment, we don't understand any of those, or we understand very few of those rules and interactions, and at a single point or across time. But if we can understand all of that, the opportunity is huge. It's just about building the knowledge.
Yeah, I think that's really interesting. The idea that the immune system and the microbiome is like interrelated, and that they influence. I think that's that's relatively new, right? I mean, this idea of
yeah, I think that's absolutely right, Annabelle. I mean, my background is clinical, and then I did an immunological based PhD, and I was a bit sort of I've come late to the microbiome, particularly through my interaction and collaboration with Sam. And, I think we used to think about these things as separate, but it's
Right.
actually impossible to do that. I think even from very early on, there's a coexistence of your immune system develops because of its exposures. We know that. And equally, I think the microbiome will evolve depending on the sort of environment that it's allowed to evolve in, which is partly to do with its own ecology and partly to do with the sort of immune and host environment that's created for it. So we can't really think about these things separately. They have to be thought about together.
And so you don't think, either of you believe that it is, that Crohn's is caused by one specific pathogen?
Sam?
I mean, I think if we come back to what we were saying before, I think it's almost impossible that every Crohn's disease patient has a single pathogen.
Yeah.
Could there be subgroups where, we're currently labeling it as Crohn's, where actually there's the presence of a particular species that's triggering one of these events that Ed was talking about? Absolutely, that's plausible. And I think that's what we really need to get to the level of understanding. But the problem or the challenge here is that will you pick that up, for example, with a fecal sample after the sort of presentation with the disease, or is that an early event that then triggers a cascade of immune response? And so it's coming back to understanding the community structure and saying, well, this is from the computational models, from the experimental systems, this is likely to be the first trigger event. So you can imagine you could have a twofold approach. If you have a risk for Crohn's early on in life, you try and manage the exposure to those potential risk factors. And then if you've got a condition where you've already presented with Crohn's and that we predict or we think that species was the trigger, you then try and set up community states of the microbiome that are very resistant to colonization with that detrimental bacteria. So, I mean, you've got to remember, it's unlikely that a single bacterial cell is floating through, and that's it floating through your gut is the event that causes the disease. It's much more likely that it has to colonize, it has to establish itself. If you imagine the single cell as the seed, and then it has to grow into a community, and you have to have a forest before the disease actually manifests. If you can do anything between that seed arriving and the whole forest being established, you've got a chance of reducing the severity of the condition. But also, if you understand that there's a forest there, there's ways to remove it and then put a different forest state in there that's not detrimental. So there's sort of three stages of intervention that have opportunity, but fundamentally they're all unified in that understanding of how the community interacts with the immune system and how that happens over time.
This is good. This is very helpful. Yeah. Kathleen, do you have other questions?
Those are all the questions I had. Thank you both so much. This was a really helpful topic for me to understand as a patient. I think from our end, we see, again, things happening so slow and we get really excited about these new therapies. So understanding where the gaps are and how we can connect the dots and that it is happening is really encouraging.
And Lisa, do you have any questions?
I think you covered them all. It gives us some hope that probably in the next decade, maybe we will actually see some new, safer treatments that may be effective for our kids and for you, Kathleen.
Yeah, thank you all for your time. I think the other thing to say, just to bring it back, is we have to remember that it's only been in the last year that microbial therapies, so fecal transplant, have had market authorization. So we talk about FMT and things as though they've been going on for a while. They're coming to market now. The next phase of these targeted interventions is where we really have opportunities. But because that first microbial therapy to market milestone has been reached, suddenly the opportunities now come for understanding how those type of interventions are affecting the microbes, and then refining that for particular patient groups or for the delivery perhaps, or the other interventions that they're combined with. So my message would be, fundamentally, there's huge opportunity in this space. as with all things, we're limited by knowledge. But the effort now is really about filling those knowledge gaps such that we can have the things that do make it through. It's not easy to take something through the phase clinical trials to get to a market, but the higher quality science that's going into the identification of those key microbes that are taken through those trials, or those key interventions that are taken through the trials, the better the chance that they have success. And the thing I would say from a patient perspective is all of the detail, all of these trials that are going on, the more that people can participate and the more the detail is so critical, even if it doesn't appear that it's going to be important, if there's surveys and all the details they're asking, that whichever researchers you're interacting with are asking to collect, there's a reason that they're asking to collect that information. And every bit of detail can be so useful in understanding why a particular species or a particular change in whatever the study is doing can be so important to the overall picture. In some ways, it's just like solving a crime where the tiny little detail is the thing that makes the difference. It's the same situation in these types of research projects, where getting that real fine level of understanding is so dependent on the patients and the participants in the study being involved and helping, because ultimately the researchers can have the perfect model for how it works, but understanding the true experience that a patient is having and making sure that matches with what the computational or experimental models are saying is absolutely essential, because any one patient or any set of patients may identify a weakness in the model that then saves a huge amount of wasted effort in the research side. And so it's really bringing, and it's what your group obviously is doing, is bringing these the two groups together in such a way that the opportunities to identify those weaknesses exist, and such that we can all work together in finding better ways to treat and manage the disease.
I'm really glad that that FMT, because when my son was first diagnosed, it's something that we were talking about. I remember talking to him about it, and we were thinking, if we could only clear out his intestines entirely, like with antibiotics, and then have a fecal transplant, maybe that would be the key. And then this was back in 2014, 2015, and the few trials that were happening at the time, they all came back, well, it was sort of okay, but for Crohn's, it's not really working. And I sort of put that kind of aside. I sort of thought, okay, well, I guess this isn't the answer for Crohn's, but I'm really glad to hear that people just they're continuing on, that those results initially didn't stop people from pursuing that path, and they just needed to figure out why it wasn't working and maybe modifying whatever they needed to modify. So
yeah, I think that's exactly right, Annabelle. And I think the other thing about FMT that we've understood, apart from it being, more successful initially with ulcerative colitis, is that I think they're now calling it fecal microbial transfer, because it's not, the word transplant can sometimes imply cure, whereas it's become increasingly clear that it's a treatment that needs to be done in an ongoing way, because of this desire for the body to return its microbial state to at least something similar to before. But nevertheless, while that may be disappointing to patients out there, it does help us. All of this is really helping us understand these interactions, which is helpful for all patients with inflammatory bowel disease. And I think it does give us, as Sam was sort of alluding to, it really gives us a sort of exemplar of how we can modify the states in a positive way. So it really is a cause for hope, even though it's not the cure that many people would have perhaps hoped for.
Dr. Giles, do you think that the reason that only 30% of Crohn's patients find it successful to have FMT, and maybe that the trials are too short? I've read some papers where maybe they've only done one FMT. Do you think a longer course of treatment might yield better results?
Yeah, I think that, the 30% figure is often quoted for the UC patients, I think, but I think that, and that's sort of similar to a lot of the initial drug trials for these drugs that we know are better than that. So often in the initial trials, you're looking at patients who are really relatively desperate. Now, that's not necessarily the case. So more severe cases, more treatment-resistant cases. So that can make the initial numbers not look as good, but it clearly is telling us that this isn't a treatment for everyone, or at least not in the way that we understand it. So I think really what that's saying is that we don't, we're taking a very sort of broad approach where we're, often preconditioning with antibiotics to kind of get rid of everything that's there as best we can, replace it with something that we think is healthy because it's in a healthy individual or a group of healthy individuals. And that is working. And so that is telling us that we can change that relationship. But I think it's more of a platform to build on rather than anything else. So I think that whether it's duration of treatment, I'm not sure. I suspect it's more likely to be that it's just not quite the treatment in the right way. Because I think the treatments that do work in Crohn's can work quickly. So it's probably more, we're not selecting quite the right way of doing that intervention. I know there are groups that are looking at different approaches and to try and refine that, but we're all targeted in slightly different ways. Is it the way it's being delivered? Is it the frequency? Or is it just being more targeted in the understanding of that microbial and host interaction?
Gotcha. Thank you. That makes sense.
Well, thank you both so much for taking your Monday morning out to talk to us about it. And I think we feel very, very hopeful for after what you've said. It's wonderful. I know there's some great research coming out of Australia, not just regarding this, but in other aspects too. So it's really, it's great. It's great to hear, and it gives us a lot of hope in the community. So thank you once again. We really appreciate it.
Our pleasure. My pleasure, certainly.
Thank you so much.